All right, so I am here as sort of a representative of a biologist who has encountered some high-performance computing issues, and I have made some limited steps to address the specific problems I had. So hopefully this will give you some idea of the sort of situation where one of these issues can arise, where you really start to be limited by computing power, and some of the things you can do in R to improve it. I put a lot of work into making the improvements that I did. I still don't think they basically got me from completely crippled by speed to not completely crippled by speed, which I guess is an improvement, right? I suspect that as I discuss things with people at this meeting, there will be additional ideas that could improve this issue more. But I'm going to I'll try not to take the whole time since we have a lot of things on the schedule, but I'll try and just give you the science talk for 15 or 20 minutes just to introduce the problem I'm interested in and why it might be interesting to people, and then talk about how it pretty rapidly gets into this computational issue, and then a few of the things I did to improve it, and then Drew and others will give us more advice later on about ways to improve things. So I'm talking about statistical model choice in historical biogeography. And we should first talk about what is historical biogeography. It's really the study of how species got to be where they are. So you know where things live right now, and you know there's classic observations that closely related species tend to live in similar geographic regions, at least on a global scale. And so there's some indication that there's an evolutionary history to the geography of species. And so we'd like to be able to reconstruct or estimate the history of where things lived and the processes by which they got distributed around the planet. So that's what I'm interested in. Just to give you one example, this is some genetic data indicating basically the history of humans in the last 100,000 years or so. If you look at genetic similarities, basically you get this out of Africa hypothesis where most of the genetic diversity in modern humans is in Africa, and then you get basically a distance effect where the further you get from Africa, the more limited the genetic variability gets. You can see this reflected if you just do a sort of genetic distances between human populations. You can actually see the spatial structure of the continents reflected in the genetic structure of the population. So if you look at these two things and you rotate the map a little bit, you can basically get America being one side branch, the Australasia, Oceania being another branch, and Africa being at the other end. So it's just an indication that the geography and the genetics reflect each other. In this specific case, this is a very recent case. This is a pattern that got established just in the last 100,000 or 200,000 years. You can look at the totally opposite end of evolutionary biology and look at this was a study that took megatrees of amphibians, mammals, and birds, and tried to classify, sort of update Alfred Russel Wallace's zoogeographic regions of the world. So if you're looking globally at animals, how can you sort of develop a number of fairly discrete geographic units that characterize the most of the biogeographic pattern? And this was done by Wallace back in the 1800s, and using these megatrees, this research group decided to update that. And again, they basically calculated phylogenetic similarity and distances between each of these regions, and they actually had a lot of different regions, and they did clustering algorithms to try and find the optimal classification. And what's interesting, that you can plot all of these distances in a non-metric multi-dimensional scaling algorithm, and you can sort of get the relative distance of all these regions from each other in terms of the phylogenetic similarity of the fauna in those regions. And again, if you sort of rotate the rotation is arbitrary. If you rotate their distances matrix, you can basically get the same sort of pattern in the geography as you get in the phylogenetic distances. So this is more evidence that regions that are close together tend to be more closely related phylogenetically. You can see some moderate exceptions to that. Australia, of course, is a long ways from everything else, and that's because Australia used to be much more isolated than it is now. And there's still, Wallace's line famously goes right through here and separates the Australasian and the Asian fauna. So, this gets us right back to Darwin and Wallace, the pioneers of evolutionary biology. And Darwin had this famous saying, the more nearly any two forms are related in blood, the nearer they will generally stand to each other in time and space. And so some of the first evidence for evolution in the first place was this observation that species that look similar tend to, on average, be closely related in space. But if you go through the history of biogeography, a number of contradictions to this. So that was a big sort of advance in historical biogeography. Like, okay, we can finally explain this geographic pattern. Evolution is the explanation. But there were some exceptions that then became really interesting once you have this pattern. And one of them was the southern floras, for example, the southern beech trees is one example, have these highly disjunct distributions where you will get, like, across the Pacific Ocean, you will find these southern beeches distributed in southern South America, in the mountains of Australia, in New Zealand, in the mountains of Indonesia. These groups are very similar. It's classically the same genus, but it's distributed on basically opposite sides of the planet. And there's a number of examples of this, and this led to a pretty famous set of debates about how do you explain this sort of observation. If it's true that in general things, it makes sense that species that are closely related would live near each other because they must have had a common ancestor species that lived in one typical species range. But if things are widely split apart, what's your explanation? And there were two families of explanations. One was dispersal, which was Darwin's favorite explanation, and this was just the idea that sometimes you just get lucky, and over millions and millions of years, occasionally species will find a way to cross even large oceanic barriers. So Darwin speculated on things rafting, on vegetation rafts. You could talk about storms blowing seeds a long distance. You can talk about birds dispersing seeds. These explanations work well or less well depending on the specifics of the organism in question. But if you get really creative, you can have a dispersal explanation for anything. So one speculation that Darwin even floated was maybe there's a glacier somewhere in the southern hemisphere, going down a mountainside, and some trees are living above it, and there's a little avalanche, and the trees and their seeds fall on the glacier. And the glacier goes out to the ocean, and then calves off an iceberg, and the iceberg floats across the ocean, and then there's a tsunami or something, and lifts the iceberg up off the beach somewhere where the seeds can colonize a new continent, right? Is that insane scientifically, or is that plausible? That's an important question, right? So people who think that's insane, right, they tended to like this vicariance explanation. And vicariance means you can see the idea is that there was an ancestor that was living in both areas when they're currently split apart, and then some barrier arose later on and split up the species like this. And this explanation has also been popular. And this debate between dispersal and vicariance goes way back, right back to Darwin and Wallace. Like, back before we had plate tectonics, the vicariance explanation was couched in the form of land bridges. So people would, and if you go back to the literature between the 1800s and about 1930 or so, you'll see people postulating maybe there was dry land connecting continents back in the past. Maybe this is how you can explain how similar species are on opposite sides of oceans. And there was some evidence for this. In classic pre-plate tectonic geology, there was evidence that continents had risen and fallen with respect to sea level. There are certain cases like, the Panamanian Isthmus there that really is a land bridge, and there really have been faunal exchanges across that. So there's some plausibility to the idea, but it got to the point, if you, even going back to Darwin and another of his colleagues, Hooker here, if you look at where people were postulating land bridges, they were getting postulated sort of to connect every possible continent, right? And Darwin, this is a letter that Darwin wrote to his friend Hooker, and he's sort of ranting and raving about people postulating land bridges everywhere. And this is a great quote here. Darwin says, I must cease and try being rabid, or I must try and cease being rabid and try to feel humble and allow you all to make continents as easily as a cook makes pancakes, right? So he's criticizing people for being unparsimonious and just postulating land bridges everywhere. So if you're a biogeographer, traditionally you're on the horns of this dilemma. You could either postulate land bridges, or you could postulate these wildly improbable long-distance dispersal events. A solution seemed to present itself in the 1960s with the acceptance of plate tectonics, because now, instead of having to postulate geologically implausible land bridges, it became apparent that actually the continents themselves were moving, and your land bridge was actually the continents used to be connected back in time. And so this seemed to present a good explanation for things like these southern beaches, if Antarctica used to connect Australia and South America and New Zealand, for example. And this became the dominant sort of paradigm in historical biogeography. And this is, if you go to zoos and museums, you will see this classic thing everywhere. I've seen it in art displays in San Francisco. I've seen it, children's games, whatever. It's satisfying in the way that putting a puzzle together is satisfying, right? It's like, yes, we have the explanation. And the only problem is it might be wrong. In the last 10 or 15 years, a lot of molecular phylogenetics has come online. And one of the dominant conclusions from this is that when you do phylogenetic dating analyses, where you take fossils to calibrate a molecular tree and you put ages on the divergence times of various species, many of those divergence times are much too young to match up with the divergence, with the splitting apart of the continents. The continent splitting apart typically happened 60 to 150 million years ago or older. And many molecular phylogenies for many groups, including things like the southern beaches, it looks like many of those divergence events are just too young to be explained by rafting on the continents. So one solution is to disregard molecular dating evidence, but it seems pretty hard to just systematically dismiss that. The other solution is to go back to long-distance dispersal, even if it seems improbable. And so Alan de Quiros is one of the people who's led this charge. This was an article from 2005, The Resurrection of Oceanic Dispersal. And there's a number of articles promoting this point of view. But there's people who don't like this, people who are sort of raised in the tradition of vicariance biogeography. And there's some great lines here. The resurrection of dispersalism is reactionary and with little or even no empirical foundation. And I love it. I love the word reactionary because that's the sort of word you would hear with communist rebels against the the capitalist pigs or something. And if you were against the counter-revolutionaries, you would be against the reactionaries. And it's great language. So this book, Molecular Panbiogeography of the Tropics, was published by Michael Heads with University of California Press, sort of across the street almost from the integrative biology building where I worked. This was published in 2012. And this guy has a whole huge book analyzing biogeography from the perspective of vicariance. And he says this long-distance dispersal stuff is just not going to be part of his analysis. He says most biogeographers, accept that you could have allopatry, species in different locations by vicariance or by founder dispersal, but only vicariance is accepted here. So he just says straight up, we're just going to do it one way. Vicariance is the explanation. Another great quote here is, founder dispersal is controversial and may not exist. So I'll try not to take too long on this. This is my summary of this debate over the last 150 years. It's kind of like a movie that just has endless repetitive sequels, and one side wins and the other side wins, and it comes back. So when I came to biogeography, I thought, I was aware of this debate, and like most people, I was like, this is an old, boring, annoying debate. And the right answer is both. There's some sort of annoying compromise thing like that. But really what I thought would be better is if we could subject this to statistical analysis, right? Ideally, we could do a statistical inference rather than just picking one of these sides or doing some sort of eyeballing of your question and saying, oh, in this case it looks like this, in that case it looks like this. So the way you could do this is with an actual inference, phylogenetic inference method. So the way these methods work is you take a phylogeny, you have a dated phylogeny, you get some geographic range data for each of the species in the phylogeny. You assume some processes that allow species to move around. You conduct inference using those processes. And you can be parsimony or maximum likelihood or Bayesian. And actually, this might not matter quite as much as people sometimes think. What really matters is which processes will you assume, and then profit at the end somehow. But this assuming process is the important thing. So I'm going to take you through the assumptions of a bunch of the different methods that people use. So if we think about a phylogeny and the processes by which a species could move around, some of the processes are anagenetic. Those are processes that occur along branches of a phylogeny. And then there's cladogenetic processes that occur when a speciation event happens. And you could imagine when a speciation event happens, a species could break up the geographic range. So you could have simultaneous speciation and a change in the geographic range. So there's a lot of different, you can sort of go through the list and talk about different sorts of events. You could have a dispersal event where the range expands like this, one area to two areas. You could have an extinction event, or really local extirpation where it contracts. You could have a range switching event Nobody really believes that this exactly happens, right? Because it sort of imagines that this species just picks up and moves and has no ancestors. But actually, if you use a standard traditional character model in phylogenetics, if you apply that to biogeography, this is what you're assuming. And this is actually still a dominant method. Yeah?
That matrix, that represents the genetic space?
Oh, I'm sorry. These are areas. So these are 4 areas, right? So if you, so if you imagine and simplify your areas down into 4 boxes, if you live in one area, you live in that blue dot. If you live in 2 areas, you live in these 2 blue spots here, right? So this is a range of 1 area. Over here is a range of 2 areas. You could have a range size of 4 areas, right? It'll become a little clearer if we look at a few more. Here, so these are geographic ranges, right? So this is sympatry, where a species splits within a certain region. So here, the ancestor lives in region A, right, or the upper left corner here. It lives in this region of the map. And it's both of the daughter species live in that same region, right? So does that make sense? This is one way. You could have a narrow sympatry event. You could have a widespread sympatry event. This is debatable whether this occurs, but it's possible. Species could live in all 4 areas in your imaginary analysis. And all these analyses assume that geography is discrete. This is the way we construct these models. That may or may not be the best way to do it, but that's the way you can do these sort of likelihood methods. So here the species splits into 2, and both daughters occupy a widespread range. You could have a subset event where one of the species occupies a subset of the range of the ancestor. So there's a little subset species in yellow. You could have a vicariance event. This is where the range splits due to some barrier arising, for example. So this would be a narrow vicariance event where one daughter has a range of area 1 and the other daughter has a range of size 2. You could have a widespread vicariance event where, like, you know, a range of 4 areas splits into 2 and 2. Or you could have this sort of founder event, and this is the one that I'm kind of interested in, where one of the daughter species jumps to an area outside of the range of the ancestor. So these are all the processes. There's probably other ones, but these are the main ones that people have invoked in various conceptual models and sometimes in computer programs. And to make a long story short, there's a bunch of different programs people use, and each of them assumes some of these processes and not others happen. So these are some of the common methods. DEVA is a method that people use. DEC in the Lagrange program is the main one people are using right now. DEC means dispersal, extinction, cladogenesis. So you can see there's check marks by dispersal and extinction. And then it allows narrow sympatry here. It allows subset sympatry. It allows narrow vicariance. It doesn't allow, for example, widespread vicariance. It doesn't allow founder event speciation. So I wrote an R package called Biogeography. And the nice thing about it is that it takes each of these processes and assigns a parameter to it that controls the probability of that process. And then by setting these parameters to 0, you can turn those processes off, or by setting them to be free, you can estimate them using a likelihood or Bayesian method. So the interesting thing then is you can start to actually compare the different methods people have been using and see which ones, which models, which processes are actually important in explaining the data. So I mentioned these, issues: vicariance, dispersal, range expansion, contraction. Founder event speciation is the one that I find most interesting because it's been left out. If we go back, it's left out of all the traditional models, probably because most of these programs are written by people who were trained in the old vicariance biogeography tradition, which was dominant, until the last 10 years or so. So just talk about founder event speciation for a moment. This is where a rare dispersal event sort of instantaneously establishes a new lineage. You can imagine, a rare colonization event where some plant seed gets out to Hawaii, for example. And that only happens maybe once every million years. And so a new population is started, but it's instantaneously genetically isolated. Whether or not you want to call it a new species right away is kind of a subjective thing, but genetically speaking, it's isolated right away. And my question is can we see this in our phylogenetic data? If you talk to island biogeographers or even in population genetics, these founder event processes are actually thought to be important. So it's interesting that in the historical biogeography literature, it really has not been included in historical biogeography models. All right, so we hit this. Some people don't like founder event dispersal, but we're going to try and test this. The way you just program this is that here's a bunch of these possible events where the ancestor range is at the bottom here, and at a splitting event, you get daughter ranges at the left descendant and the right descendant. So, and if we look at the Lagrange program, which is the one everyone's been using, the gray boxes are the events that Lagrange allows. And if we want to add founder event speciation, we just think outside the box a little bit, and we just allow a daughter species to live outside of the range of the ancestor. And we assign probabilities to that using a parameter, and then we have a parameterized model that includes founder event speciation. And by setting that parameter to 0, we reduce to the old model, which is the one in the Lagrange program that people have been using. So here's another way to see that. If we have the ancestor range at the bottom before a split on the phylogeny, here's various possible ancestor ranges in a 3-area analysis. And then we have the left daughter has all the different possible ranges, and the right daughter has all these different possible ranges. And so you have a combination of, the number of ranges here times the number of left possible descendants and the number of right possible descendants. So you have a large number of combinations of ancestor, left descendant, right descendant ranges here that you have to look at. But you can assign parameters to each of the types of events, sympatry, subset sympatry, vicariance, or we can add J to represent jump dispersal, and we can put those events in there and give them a weight and calculate the probabilities from that. So this is what I've done in Biogeography, with this weight parameter J. And yes?
What are all the empty cells?
Those are all zeros.
Those are impossible?
Well, it's, for example, if the ancestor is A and the daughter is going to be CC, right? The daughter species will be C and C. Does that make sense? So you're making some decisions ahead of time. It's just going to be impossible for the daughter, or for the ancestor to be A and the 2 daughter species to have range area C, right? Like, maybe that's real, but probably not, right? So we just, all these blank cells get a probability 0. So a lot of the imaginable events we're ruling out. You can already see, by the way, in terms of computational issues, so this is just, we have a system, this is like a 3-island system, right? We have island A, island B, island C. And even with just 3 islands, the number of possible geographic ranges, right, is, what is that, 7. And then you have 7 times 7 times 7 combinations that your computer has to search for and assign probabilities for, at least in the naive programming of it. So imagine if you had more areas and even more possible ranges, and then even more possible combinations of left and right descendants. It gets slow very quickly. All right, so I use 3 R packages to try and implement this in the speed at which it will run reasonably. The first one is called RexpoKit, and this is just an R wrapper around something called ExpoKit, which is a Fortran library developed by Roger Sigi, and it uses a Fortran method to do matrix exponentiation. And matrix exponentiation is what you use to calculate the probability. Let's say you know the ancestor state here, you can calculate the probability of any possible state at the top of a branch. And it's just a Markov chain, and you calculate transition probabilities given the length of a branch in time. And normally, that is the core of all these, DNA phylogenetic methods, amino acid phylogenetic models. And normally it's very fast. If your matrix is only size 4 by 4, like 4 DNA, you have 4 possible states in the ancestor for DNA, and you have 4 possible states in the descendant. That's a very fast calculation. But let's say you have 500 different geographic ranges that are possible. Then you have 500 possible descendants. You have a transition matrix that's 500 by 500, and that starts to get slow. So my attempt to speed this up was to, as people recommended, to take those slow steps, which get very slow in R, and send that matrix exponentiation to a Fortran package. And it was actually a lot of work figuring out enough Fortran to even get the right inputs, and the right outputs from it. But once you have it working, it's actually pretty good compared to the other R matrix exponentiation things, especially for big matrices. So this is RexpoKit, and that was an adventure in merging Fortran and R. And then to do this cladogenesis part of the calculation, where you have an ancestor and you have descendants, 2 possible descendants, this involves, you have to search through, starting conditional on some ancestor, you have to search through all the possible combinations of left and right descendants and assign probabilities to those, conditional probabilities. And so you're not doing a matrix exponentiation, but you're just literally going through that list. And this is a for loop problem, because you have at least 3 nested for loops to go through that. And that also got incredibly slow. Like even with maybe 4 areas, it was noticeably slow in R. So here I used RCPP to do those nested for loops, and I got a dramatic improvement. And also when you're programming something, because of all those combinations of ranges, ancestor-descendant ranges that are 0, if you're sort of smart about it, you can exclude a lot of those ahead of time, and you don't have to go through the entire combination of everything. So I have an R package that does that, part 2. And then it's all stuck together in this R package Biogeography. And these packages are all online, so if you ever need, and I'm kind of an idiot, especially when I started this, right? So if you want to see like a beginner figuring out how to use RCPP and doing like the simplest possible things like sending stuff to a for loop and getting it back, you can look at the Plato RCPP code. Or if you want to see how to call a Fortran function, you can look at the RexPokit code. That might be useful to you, because it's pretty opaque. Like even if you know R, getting these other things to work is non-trivial when you're starting out. All right, so let's talk about some of the results of this. There's a lot of other features, and you can put distance matrices in and make dispersal probability dependent on the distance between regions in this biogeography analysis. You can put in the size of areas. You can do time stratification. You can put in fossils. There's a lot of other features I added. Basically what I was comparing was my program to Lagrange, which is the previous program that's widely used, hundreds and hundreds of citations. And if you want to try this out, you can go to this website. I've got an example code that you should just be able to paste it into your R. After you install Biogeography, you should be able to paste it into your R window and run an analysis. Here's sort of the example data set, Hawaiian Psychotria. This is a shrub from Hawaii, and it has this sort of classic pattern where a lot of the early diverging lineages live in Kauai, which is the oldest island. So the Hawaiian island chain is a series of volcanoes, and the oldest volcano is Kauai, and the youngest one that's still erupting is Hawaii. And so often when you look at phylogenies, you'll see the oldest branches being in Kauai, and then you get a series of colonization events as you move to younger islands. And you do see that in sort of a Lagrange analysis. But what's interesting is if we take the Lagrange program, which is the one that it just has dispersal and extinction, you get this log likelihood out of it. You calculate the probabilities of the data, you get log likelihoods, you get -34. And when you run that same 2-parameter model in my program, you get the same log likelihoods, which is nice. So I'm replicating, completely independently programmed to get the same log likelihoods. And in a lot of different analyses, I get the same answers, which is good. So I successfully replicated this. But then we can compare the 2-parameter model in this Lagrange program to my 3-parameter model, where I've added a J parameter controlling the weight of jump dispersal at cladogenesis, which has an implicit 0 in the Lagrange program. So when we do that, if we compare that 2-parameter model on Hawaiian Psychotria, we have -34 is the log likelihood. When I add this parameter for J, I get a positive value for J, and I get a log likelihood that's -20.9, which is about 13 units higher than for the traditional model everyone's been using. Also, the D and E parameters, which control range expansion and range contraction, they basically go to 0 for this data set. This doesn't always happen. And J jumps from 0 to a positive value. So if you do your standard model choice statistics, you can reject the null model, that these are two equivalent models with an extremely strong p-value. You can calculate ASCs and get a relative probability of the two models, and you get 99.9% weight, on the newer model, like I said. And for adding one free parameter, I consider this a pretty good result, right? Like, it's not bad. And you get different parameter inferences. Is this just a one-off? Was this just this specific data set, or does this apply to other data sets? So I applied this to a bunch of other data sets. Here's Lonicera, honeysuckle sertandra is a shrub in the Pacific. And again, if you look at the relative probabilities of the DEC plus J model, the 3-parameter model, it ranges from overwhelming support to strong support for the DEC plus J model in these kinds of island systems. And this is where you start to get worried, actually, right? Like, because if you're always doing better in your likelihood model, maybe either you programmed it wrong, or maybe there's some artifact where there's some bias in favor of your model. So I was paranoid about this because you don't want to come out with something and then turn out to be wrong about it, right? So, you can test this, though, by simulating under the parameters for these different models and then doing inference under those models. And so when you do this, if the truth is the DEC model, the two-parameter model, and then you do inference with dec plus j, where you're adding another parameter, you always do a little bit better with an extra free parameter. But the amount that you do better here, this is the log likelihood advantage of dec plus j over dec. The amount you do with adding that free parameter is 1 or 2 log likelihood units better, which is actually exactly what you would expect for adding just a free parameter that's not in the simulation. So you only do a little bit better. And if you do a likelihood ratio test, you only reject the simpler model 5% of the time, which is what you hope if your p-value is 0.05. However, if you simulate under the dec plus j model, the inference you made for the dec plus j model, and then you infer under dec plus j and dec, the advantage of dec plus j is now up to 10 or so, 10 to 15 log likelihood units. And this is exactly what we see in the empirical data. So it looks like reality looks more like it was simulated under this model than under this model. Now this is just one example. Due to reviewer comments who still didn't believe it, there's a lot of other things that could be going on. The phylogenies, molecular phylogenies, are missing species because things have gone extinct or weren't sampled. You could have state dependence in your speciation extinction process. So it could be that large islands have higher speciation rates and lower extinction rates than small islands, for example. And there's a lot of possible parameter combinations of these parameters. So I did simulations under all these different combinations. And here is the basic chart. If we look at, these are D and J, the two main parameters here, and the true values are these black dots. And then inference under the two models, the inference under the two-parameter DEC model, dispersal extinction cladogenesis, is the white boxes, and inference under the DEC plus J is the gray boxes here. And so you can see in a case, and so I'm comparing them side by side. So this is DEC, DEC plus J, DEC, DEC plus J. And you can see in a case where D is 0, but J is positive, the DEC plus J model, nails it. There's like, yes, there's a positive J. The DEC model has J fixed to 0, so there's no way it can get J correct. And instead, it assigns those events to this D parameter. So you get D raising artificially. However, you can also have a mix of both processes. This is the range expansion process and the jump dispersal process. And you basically get both of those processes inferred correctly, so that's good. If you look at when you reconstruct the ancestor ranges on the tree, you can look at the proportion correct. So again, white is the two-parameter model, and gray is the three-parameter model. And in cases where J is an important process, you really do get, especially as J increases in value, you get much higher accuracy if you put J into your model instead of not having it. And you can look at the delta AICC values between the two models. And again, when J is an important process, you get huge differences in the models. So this is under a tree where you're assuming that, this is under simulations where you're assuming speciation and extinction don't matter. You can do a complex simulation where here the probability of speciation is a function of range size. So, and extinction is also a function of range size. So if your range is size 4 areas, your probability of speciation goes up by 4, and your probability of extinction goes down by 4. So this is a pretty biased state-dependent model, and if we compare the two back and forth here, you get a little bit of degradation in your inference, but really it's not overwhelmingly bad. You get some hit to your accuracy, and your model choice changes a little bit. But overall, you can still tell apart models where J is high from models where J is low, and you can distinguish these models. So I concluded that this really does work pretty well, even if you have a more complex simulation model than anything in your inference model. These as long as your rates of change, you can see there's a degradation when you start to get super high rates of change. Your accuracy goes down. That's over here, but that's almost the case with any phylogenetic method. If you have high rates of change, it gets harder. You lose your phylogenetic signal more quickly. So anyway, so I think this really does work pretty well. And you can see that even with these complex models, like leaving out state-dependent speciation extinction, it matters a little bit, but it matters a lot more leaving out J in cases where J is important. You really get a big hit on your accuracy in your inference. So I think we should use this founder event speciation model, especially in island cases at least. This is basically what's going on. If you have founder event speciation, you have a pretty simple explanation. If your species are each living in single islands, you basically have a, you have a jump event at speciation and you're done with your explanation. Whereas if you force a model without that, then you have to have these range expansion events, and then you have to have waiting times where nothing changes. And every waiting time in a phylogenetic method decreases your probability. It decreases the likelihood. So I could put these on. So you have all these waiting times where you have to have some positive value for the range expansion process, for example, because you have to get something from A to B. But if you have a positive value, then waiting and having no changes becomes improbable. Whereas here, you just have an event at speciation. And these range expansion processes often zero probability. So if there's a zero probability of a change along a branch, the chance of having, it's a high probability that you will have no changes on the branch. And so that's the advantage of this model. So anyway, there's a lot of cases where this matters. If you compare the inferences you get for this Hawaiian Psychotria clade, this is the inference you get under the standard DEC model, unconstrained. And you get this inference of a lot of widespread ancestors, because the way the model explains geography is things spread out and then break up. Whereas with the founder event model, you just get this sort of history where, just compare back and forth here. Right? Founder event model, you just have a few jump events is all that's really necessary to explain the distributions. And if you talk to island people, right, they're like, this is what we always thought. And so it's just somewhat surprising that the models we had sort of forced this kind of answer. I think it goes back to that history of biogeography. And also your uncertainty in your estimates often decreases. This is the DEC model. This is the 3-parameter DEC plus J model. Often, it's not 100%, but you get a big improvement in your confidence in some of these answers. And we did accuracy already. Your accuracy is better. And I didn't just run this on a couple example clades. I ran it on 13 different island clades. And this is basically the ratio of the AIC weights on a log scale. So depending on the size of your data set, you can get up to billions and billions of times more probable, the DEC plus J model being more probable on a big phylogeny. And that's just the multiplicative nature of the likelihoods doing that. So anyway, almost every data set overwhelmingly supports the DEC plus J model when they're island data sets. It's not always the case. We should skip ahead here. Yeah. Well, so this is the thing. So I think this is the danger of having models that have fixed parameters. So the DEC model, which is the widespread, it was a revolutionary model because it used a likelihood analysis to do biogeography, and it was a probabilistic method. But they had a fixed process in there that you couldn't change. They had 2 free parameters, but there was no way to do this cladogenesis process with free parameters. So once we make them free, we can start to discover improved models. And I just consider the model that I've created here just one step in this. There's a lot more. I showed you a number of other parameters that still have hardly been explored. And I think this situation is a lot like the DNA substitution situation, where the first model people had was the Jukes-Kanner model of DNA substitution. This just assumed equal transition rates between all possible states. And this was a useful model, right? It really, it captured, for example, the phenomenon of saturation, where you get repeated substitutions and you start to lose phylogenetic signal. So this is a really useful model to have. But people started adding additional parameters to it, and you ended up with, Chimera 2 parameter. You got, and eventually you have the GTR, general time reversible model, plus I plus gamma, for example. And so I think we should do the same thing in biogeography. So another implication, all models are wrong, but some models are useful. And I think we need to actually do estimation in biogeography to figure out which models are wrong and not useful and which models are wrong and useful. All right, so just to show one case, there are some empirical cases where you do not support the founder event model over the traditional model. This is one. They tend to be continental clades that are not on islands, and they tend to have a lot of, if you look at the tip geographies here, there's a lot of overlap among the species, and this is the kind of pattern that you expect if the DEC model without founder event speciation is true. If you add founder event speciation, you get a lot of basically single area endemic species. So if you have a lot of overlapping species, you may support the traditional model. If you have a lot of single area endemics, you might support the founder event model. So these are Tagetes butterflies. I have the beer here because I had a beer challenge. If anybody found a clade that supported the traditional model over my model, I'd buy them a beer. But I found this case myself, so I bought myself a beer. All right, so founder event speciation is important almost everywhere, not completely everywhere. Many data sets falsify the current models that are currently in high use. I've tested some other models, but we don't have time for that. And with Biogeography, we can test all these models. So I got one point, and I'll talk about computation for a second. If you're interested in this general issue, Alan DeKiros, who was one of these guys pushing the sort of reemergence of long-distance dispersal, he has a popular book that came out just this year in January called The Monkey's Voyage, and it's a really great book. It's the best book in biogeography probably since David Quammen's Song of the Dodo. And he talks about this vicariance dispersal debate. And I was pretty gratified. I came out with this PhD that had the conclusions that I showed you, and then a few months later, this book comes out. It's like, yes, that's exactly what I was trying to say. So this guy says it much better than I do, but I have the statistical part of it. So I think that's good. And it's great, especially he goes and he interviews all these different scientists, that some of them who say these things about reactionaries and dogma and all this stuff. And so it's great to get the human side of the science. And like I said, there's the wiki page if you want to try this, or even just look at the code. I should talk just a little bit more about the calculation issues. The thing that really kills me is the fact that if you have n areas, right? So if you have 5 areas, the geographic range is every possible combination of presence and absence in each area. So if you have 5 areas, you actually have 2 to the 5 combinations of areas that are possible geographic ranges. Right? So if you had areas A, B, C, D, E, you could just live in area A, you could just live in area E, but you could also live in area A, C, E, or whatever. And each of those becomes a different state, and so your state space goes up at 2 to the n, where n is the number of geographic areas. So if you have 10 areas, I think you have 1,024 possible geographic ranges, and that means the matrix that you're exponentiating is 1,024 by 1,024. And that takes a second or so on the computer. And if you had 11 areas, it would double again. So then it's 2024 or 2048, et cetera. And so this puts a really strict limit on how big an analysis we can do. And if you think that there are more areas than 10 in the real world for some species or some group of interest, then this becomes a real problem. There's a few tricks you can do. You can limit the maximum size of ranges. So if you say that nothing can live in more than 2 areas, then you can do more like 20 different areas. But then you put a real tight limit on what things can do in terms of how many areas they can occupy. The Rex-Bookit solution really improved things over standard R, but it still hits this limit. And probably any method will eventually get knocked back by this exponential increase. Rex-Bookit does have a sparse matrix exponentiation capacity, and sparse matrices are supposed to be sparse matrix routines are for matrices that have mostly zeros in them, and this can speed things up. I've tried with this. I've tried the calculations with this, and they tend to be kind of approximate calculations, and so I haven't pushed it very much further because especially in maximum likelihood analyses, you kind of want to see that you get exactly the same answer. Bayesian method, which I've actually got, that may be one solution. Another solution is parallelizing these computations. So if you have matrix exponentiation, this occurs on every branch of the tree, and you have to do every branch of the tree at every iteration of the maximum likelihood search. So that's what gets slow. Inside Biogeography, I've used MCL apply to farm out those matrix exponentiations to different processors. So that, if I have 4 processors, I get 4 times faster, which is, I'll take that, especially, if it moves it from 10 hours to 3 hours, that's great. I could imagine using this on the cloud or something and doing, a really big analysis, but I have not actually tried that. The other thing I mentioned, Clado RCPP, this problem of at speciation events, you have every combination of ancestors left descendant, right descendant. Searching through all those with for loops is super slow, unless you use C++. So this is a good example. The routines are actually really simple, but I got a huge improvement using Clado RCPP just because C++ for loops are way faster than R for loops. So this is something you can look at. These packages are both on CRAN, and they may give you some good examples to look at. Yeah, and this is how we put them together. So RexpoKit, matrix exponentiation in the branches, and C++ for loops at the nodes. So I think I took enough time there. I'll take questions if there are any. But this is just to sort of set up how an originally simple question can turn into a complex computational thing. Yeah?
Is it possible to make a number of areas a parameter so that it looks like dynamically determined?
You could, yeah. I've not tried that. Like, there's always this question of, at the beginning of these analyses, you have to set up some sort of postulate about, okay, this is our geography. And when you have islands, it's somewhat clear, right? You can say, okay, these are the 4 main Hawaiian islands. Those are our 4 areas. So that's a simple system. On a continent, people typically use ecoregions or something, but species do whatever they want. A lot of times there's often this question of a species just, there's 3 little populations in some other region. Do you count that as present in that region or not? So everything's approximation. You just have to make a guess at it. I do think it would be worth people testing their area hypotheses with these same likelihood methods. You could postulate a set of areas, then you could cut off one, cut off another one, and do the calculations that way. And I think you would find that if you moved to the smallest possible number that matched your data, you'd probably do better because you'd be spreading out the probabilities less over all these possible combinations of areas. But yeah, no one's really tried that yet. Even doing model testing in biogeography, like I'm sort of the first person to try this, and so, once that gets published, then we'll move to the next step. Yeah.
So lots of the parameters that you showed, like 10 times or so,
Mm-hmm.
the D, E, and J.
Mm-hmm.
Were you surprised that, I know for that first example, the D and E parameters were like 0?
I'm not, mostly because of.
It seems a bit surprising that, they're just all 0.
Yeah, it depends. It's not always like that, but let me see if I can. There was one, I think I took out the slide. This is one of them. This just has a few examples. So they often go to 0. This one went to 0, 0. Yeah, this one didn't. This only cut D in about half, right?
Do you think that's a bit odd?
I don't think so. I think in these island cases, the real process is probably founder events,
So what does
right?
So what do D and E stand for?
So D is range expansion, dispersal in the sense of range expansion, where you start in one area and you move to 2 areas. And E is range contraction or extinction, where you move from 2 areas to 1 area. And these are processes that occur along branches. So, and J is the weight of founder events at cladogenesis. So if you can explain everything with a few founder events at cladogenesis, then you no longer need any positive value for these other parameters. That's basically it, which is interesting because in the model people were using, J was fixed to 0 and D and E, right, were the free parameters. So
It's almost like it kind of actually reverses to 1 parameter problem then, essentially, like in terms of the model.
it would. You could, for this sort of data set where it really does go to 0, you could fix these to 0 and you'd have an even better model. But the problem is sometimes you do get positive weights for these, so you don't know ahead of time. And typically these ones where you get some positive probability for D are cases where, if you have 2 sister species that each live in 2 areas, there's no way for founder event speciation to explain that with 1 event. You need some sort of range expansion event on one of the lineages to do that. So when you have, and you do see this, like I think I showed you this one, in cases, like it all comes down to this. If you have this situation where everything lives in 1 area, this is kind of a classic founder event model. If you have a situation where things live in multiple areas, this is more like the DEC 2-parameter model. So if you run the DEC plus J model on this data set, you get the DEC model back, and you get J equals 0. So, I think this is great. It says, hey, our data can tell us which models are the right ones in different situations. So, yeah.
So how is dispersal actually modeled?
So dispersal, you can, the simplest case, which is most of the ones I've shown here and most of the published analyses, dispersal in the sense of range expansion is just given a probability per million years, and it's a standard Markov, rate per exponentially distributed waiting time for events, right? So it's just like the waiting time between switching from A to C in a DNA model, right? Moving from area A to area A plus B has a waiting time, depending on the value of D, the range expansion parameter. J is a weight of, there's multiple possible events at speciation, and if J equals 0, founder event speciation has 0 probability. And if J has a positive weight, then basically you sum the weights of the different events and divide by the sum to get the probability. So, and that's more time than we have to go into, but it's actually a pretty simple calculation. And it all goes back to that, way back I had a table that had all the different parameters in it for cladogenesis events. So they're just parameters that control processes like any other parameters in a DNA substitution model or something. So hopefully that helped a little bit. It's a lot to, I'm sort of dumping all this stuff out in a short amount of time, but anything else? I should surrender the mic here. Thank you very much.
Thank you.